Okay. Welcome, hello, and Happy New Year, everyone. The Total Care Program team is excited to ring in 2026 with our fourth installment of the Ask Your Doctor webinar series. Before we get to it, here's a couple of reminders for today's session. Your participation is highly encouraged. We recommend using the chat feature so your remarks can be shared with the speaker at the appropriate time. If you do prefer to speak, click the raise hand icon. The moderator will ask you to unmute when it's time for you to share your comments. Please note, this event is being recorded. By participating in today's sessions, you consent to be included in the recording that will be posted for future reference. Should you prefer not to be recorded, please use the chat feature only. We know you're excited to get started, so without further ado, here's Dr. Sood to begin today's presentation.
Hello, everyone, and Happy New Year. We're excited to have so many new attendees here today, and this was a much anticipated conversation. So we're going to start just in a bit after I make a few announcements. So I am Anubha Sood. I lead the Total Care Program. You can learn about the program on our webpage. If you register for the program, you will start receiving emails and information about all of our events. So next in the series, I just want to let you all know in February, we will have Dr. Sharma come in to talk about surgical options for PD. And then we have our big annual symposium coming up in April, on April 25th. So you will get to register for all of these. We highly encourage your feedback. So after this event, please give us feedback. That really helps us with our future events and programming. We are also launching a Total Care Program Impact Survey that will be coming to you very soon. And we would really appreciate you filling that out. Now, without further ado, I'm going to introduce you to Professor O'Sullivan. He is a professor in the Department of Neurology and the medical director for outpatient neurology services at UT Southwestern. Dr. O is an expert in the latest treatments for movement disorders. These include injections of botulinum toxin to reduce muscle contractions and associated pain, and deep brain stimulation to help control symptoms. So Dr. O is going to speak to us today about neuroprotection and current state of research. Thank you, Dr. O.
Thank you, Anuba. And thank you for participants for your interest. I'm going to go ahead and share my slide deck. I'd like to run through, I've got 6 or 8 or 10 slides. Let me see, here about 9 slides, and then we'll stop for some questions. So the conversation or the presentation today is about research about disease modification in Parkinson's disease, and that's specifically neuroprotection and neurorestoration type research. So again, the focus today, so just to comment, there is research in many topics about Parkinson's disease, including research to identify the causes and how to diagnose early Parkinson's disease, and how to get better drugs to treat symptoms like the tremors or the cognitive or other features, other symptoms of Parkinson's. There's a lot of research on better drugs for that. But that's not what I'm going to talk today. There's also testing of symptomatic relief with complementary and alternative therapies, and psychological and physical rehab therapies, and also research on neuromodulation. That includes best ways of doing deep brain stimulation, transcranial magnetic stimulation, and focused ultrasound treatments. So there's research in all those areas. But again, that's not disease modification in terms of neuroprotection or neurorestoration. So our presentation is about changing the disease course, not kind of addressing some, you could think of Parkinson's like a train in motion, and it's causing consequences along the line from the process. But and so there's research in trying to ameliorate or mitigate some of that downstream damages and help the symptoms. The research that I'm mostly going to focus today is about how to stop the train, essentially, or even slow it down. So the primary focus is research in humans. There's also a lot of research goes on in test tubes, on modeling, computer modeling, and on animals particularly mice and fruit flies or other animal on which we test things to see if they might have Parkinson's disease. But as we may discuss later in the visit, a lot of things that look promising in test tubes and animals haven't actually worked out in humans. That's been the track record of the past few decades. Things that we think, based on the preclinical research, might work in humans. So the discussions I have today about things that are actually being tried on humans that have been tried and are being tried on humans for Parkinson's. Before we launch into discussing the particular strategies and therapies that are being researched in Parkinson's disease, I just want to empower you to do your own investigation about clinical trials going on in Parkinson's disease. There's a website called clinicaltrials.gov. I'm going to link to it here and show you how to navigate through it, that I think is a very good resource for you to do your own search for what research trials are going on, what credible, well designed and well regulated with appropriate ethical review. Any human trial in the United States has to register on this website. And so it is a good resource for investigating that. And I'll show you, if you want to search, okay, what trials are going on in Parkinson's. Now, the other thing about this is research in humans that you're going to find on this clinicaltrials.gov. You may have other resources where you'll Google and find research going on animals and so on. But for reasons I just told you about, I take that with a grain of salt. Anyway, when I look on the website today for research in the United States on people with Parkinson's disease, I'm finding 170 trials are currently recruiting. There are another 23 that are approved and are ready to get going, but they're not yet recruiting. And there are another 60 that are still ongoing testing a treatment, but they're no longer recruiting patients for them. So that gives you a bit of a sense for how much clinical research is going on. About 55% of these trials that I just mentioned are for symptom relief. That's to alleviate some of the downstream symptoms like tremor or the cognitive difficulties or the mood difficulties or balance difficulties, things like that. And about the other 45% are for disease modification. And that's the focus of today's presentation, about treatments to try and slow the rate of progression or to stop it or reverse the disease itself. I put a link here, and I'm going to go to it to show you how this website works. By the way, there is a Fox Trial Finder on the Michael J. Fox Foundation .org website, and they have a Fox Trial Finder that's organized a similar way, but there's no requirement that that trials get registered there. So it depends on the Fox people curating it and managing it. The clinicaltrials.gov, I have high confidence that any trial, human trial, is going to be registered there. So I'm going to jump off this. I'm going to make a link to that. So I'll stop share this screen, and I'll share instead my browser that's going to show. And this is what this clinicaltrials.gov website looks like. It's maintained by the National Institute of Health. And I did a search and I came up with this study. This is the title study: Assess Safety and Efficacy of Surgical Implant of this RDP001 in idiopathic Parkinson's disease. So I'll tell you what's going on. It's testing safety and tolerability of this intervention implanted during a surgical procedure. It's a stem cell transplant, and it's a phase 1 or 2 study, which is an early phase trial. Research studies in humans, we've got phase 1, which is in healthy people, phase 2, which is a small, phase 1 can be, very small numbers anyway. Phase 2 is medium size. Phase 3 is the main studies. There are phase 4 studies, but they are less rigorous, let's say. Phase 2 and phase 3 are the most important clinical trial studies. So this will tell you, if you were interested in this stem cell treatment, then you'd find what locations are doing this. Where's the contact information for it? You'll see where they're looking. You can refine your search for finding places in Texas or whatever area you're interested in going to. You'll find the eligibility criteria. It'll say it'll say you have to be 45 years, 75 years, and so on. You'll get some idea of the study plan. You can expand and see more details. They won't give you the consent form here with all the risks and benefits, but they'll give you enough information that you can decide, yes, I'm interested in this, in which case you can reach out to one of the study sites, the contacts and locations, and say, okay, I'm interested in this trial. I'll be in California because I'm relocating there, and so on. So that's the a way to search for your own clinical research that you want to be part of. I'm just going to go to the clinicaltrials.gov web page here, and you'd put in Parkinson's disease as condition you're researching for. You might be interested in neuroprotect. Well, you might be interested in people within the state of Texas. You might be within Dallas area particularly, or you might be particularly interested in recruiting, not your recruiting studies. That would be a search field, and this comes up with 48 studies, and now you can list through each of them and see, and get a listing of them, and then see if that's a good study that you would want to be interested in. All right, that is a little bit of a intro to the clinicaltrials.gov resource for people who want to search for their own trials. I'm going to move on here. And is my screen, Anuba, showing the currently effective? Okay. So I'm going to talk about, new research that's coming down the pike here. But first, I should say, when we're talking about neuroprotective research, there are two things that have some already showing fairly good evidence for neuroprotective effect in Parkinson's disease. One of these is rasagiline, and the other is aerobic exercise. Now, I got to say, while the evidence is convincing enough that I consider them to have a neuroprotective effect in Parkinson's disease, the effect is fairly weak. It certainly doesn't stop the disease. It slows it down, I believe, to a meaningful extent, but not as much as you or I would want. And but anyway, let's talk about these two interventions. Rasagiline is developed as a monoamine oxidase inhibitor, so reduces free radicals, might be part of its mechanism of action. There have been multiple randomized, double-blind, placebo-controlled research trials, which are the coin of the realm, the state of the art for testing whether something is effective or not on this drug in people with early Parkinson's disease, and overall show statistically significant but fairly small benefit at 6 months to 18 months. That's the sort of time frame that these research studies, most of them are at 6 months or 12 months, one or two of that to 1.5 months. So statistically significant benefit, but in terms of the clinical magnitude of the difference, it was actually pretty small. There was a narrow error bar or margin of uncertainty about the result, but it was a small overall effect. Now, that may be because Parkinson doesn't change all that much underlying Parkinson severity over the course of 6 months or a year on average. And so it's hard. Even if you stop in your tracks, the magnitude of the effect would be pretty small. But regardless, when this company who owned this 10 or 20 years ago brought this to the FDA to get approval to market it as neuroprotective in Parkinson's disease, FDA had a split decision. The recommendation panel was almost split down the middle. I think 6 in favor of approving it for neuroprotection in Parkinson's disease and 6 against. And the ones who were against it said, yes, but the effects are too small and they're not hard enough. They're not, beneficial enough. We're not going to allow you to market it as neuroprotective. So the company essentially sold it off, and now it's generic and it's readily available. But the evidence is still there. Evidence enough to convince me and at least half the FDA panel that, yes, it is neuroprotective, albeit to a small extent. There was longer-term study following patients out to 5 to 7 years in a Scandinavian country, but those sort of research studies where you follow how people are doing, you're not randomizing, and you're not. There's a lot of dropout. Patients move, some of them die, some of them don't keep coming back. So there's a lot of missing data, and therefore there's a wide bar of uncertainty about the results. It looked like a large benefit at 5, 7 years, but there's a lot of uncertainty. So, and it wasn't a well-designed randomized controlled trial. So it kind of fits the narrative that I believe that the benefits accumulate over time, but I'm not sure about it. And it certainly doesn't convince many of my colleagues, Parkinson's specialists. It's compelling enough for me that I fairly commonly recommend rasagiline. There are some downsides to it too. We can get to in question answers if we want to. But anyway, there's that. Second thing is aerobic exercise. And we're talking about moderate to intense aerobic exercise, getting to heart rate over 100 for 20 minutes 3 times a week. That'd be a general objective with your aerobic exercise. You can do that by cycling, could be stationary cycling, could be elliptical, dance exercise, swimming exercise. Those are all very good. Boxing is probably good if you keep it up with though it's probably not, I'm not sure if it's consistent for that duration. But anyway, there have been many trials, most of them pretty small, showing that people who do this do get benefits in their mobility, and it does seem to be sustained in a way that probably suggests a neuroprotective effect. There also are research studies on people with Alzheimer's showing they improve their memory in a way that probably is consistent with neuroprotective effect. Now, I've got to say the caveat about the quality of that research is these are usually pretty small studies. And even smaller studies, you can look at brain activity, brain measures, and they can show a benefit too. But it's difficult to randomize subjects to either be active or not active. Everybody should be active. And so what they usually do is randomize patients to be more active versus less active. Now, whether people actually do what they agreed to do in terms of keeping up physical activity, you know how hard it is, unless you're really committed to keep up a rigorous physical exercise program month after month at the recommended amount. Even people who signed up for it and said that, yes, if I'm randomized to that arm, I'll do it. Not everybody did it. So it ends up, and then some of the people in the other randomized arm kind of got happy with the exercise, got better at it. So it does lead to, it's pretty hard. It's not like a drug where you can't get it unless it's provided or it's injected, let's say, and you're not going to get it otherwise. You are going to get it if you're randomized. We know everybody gets an injection treatment or stem cell treatment, and we know who doesn't get it. But exercise, people can do that if they want to, don't have to do it if they don't want it. It's pretty hard to be sure. So there are lots of caveats in the aerobic exercise. However, we still get a bit of a sense that it's neuroprotective. We certainly think it's healthy for blood pressure, bones, cardio, 101 reasons, weight, so many other things. So we recommend it anyway. So that's currently somewhat proven neuroprotective steps in Parkinson's. The main strategies that we look at currently are neuroprotection and replacement or restoration therapy. When I talk about disease modification, so neuroprotection, probably the main thing, within that you'd include biochemical adjustments. That probably includes antioxidant type strategies or GLP-1 type medications. And another neuroprotection strategy is to remove alpha-synuclein. I'll talk about that here in just a minute. And then there's replacement restoration therapies. That's trying to put back something that's gone. So if you've lost neurons or lost cells in your brain, you try and put them back. Well, stem cells is an obvious way to do that. Inject, new neuron cells into your brain. That's replacing something that's gone and try to reverse the process. Or you could inject genes that will stimulate new production of cells, for example. So they'd be kind of replacement or restoration type therapies. So those would be, generally a kind of an outline of the strategies that can be used in research. Now, among the biochemical adjustments with small molecules, that does include the antioxidant research and ongoing research in these areas. For example, resveratrol, which is in the red wine, for example, and Broxol, which is in a mucolytic, a cough syrup, that agmatine, limonene, which and morignan are sort of plant derivatives. I actually don't know a whole lot about most of these things, but based on test tube work on cell models of Parkinson's disease, each of these had a signal of benefit, and they are all relatively safe. So they're all undergoing testing in humans with Parkinson's disease, and may or may not, pan out. Certainly, none of them are established. If they were, I'd be showing them in the previous slide about the effective treatments. There are other strategies like chelating down iron. We might have put in here, there have been research on other things on the right side of the screen, things that have been tried in recent years and made sense based on test tube work or theory or computer modeling. And unfortunately, they haven't panned out. When they put to the test, when the rubber to the road, in terms of trying in people with Parkinson's disease. We haven't had good luck with GLP-1 analog exenatide, vitamin E, coenzyme Q10, N-acetylcysteine, simvastatin, or also Zocor, or Actos. There are various other small molecules that try to change the biochemistry of the nerve cells to make them last longer, survive longer, be more healthy, that seem to work in test tubes and kind of crashed and burned when they went on to human research. And I was going to remind myself, brackets here, to comment on preclinical Parkinson research. That's the test tubes in animals. So the deal is that animals don't get Parkinson's disease. So it's pretty hard to test something for changing the course of Parkinson's disease if you don't have a real good model. So what we try and do is a kind of a not very good model. We give animals toxins to try and kill certain neurons, and then we try and prevent the injury or so on by trying these molecules. And most of the molecules that I've got up here on the right-hand side of the screen were quite effective in those models. And then they didn't work in humans. And that kind of gets to the point that animals or cells don't get Parkinson's disease. Intact humans with aging under some influence, some of them genetic influences and some of them environmental influences, are susceptible to deterioration in this complex process that is Parkinson's disease. But animals and test tubes and cells don't capture the same process. So testing something there and then transferring it to human testing has just not been a very good, hasn't worked out well. We haven't had a good track record of success. I want to talk for a short while about alpha-synuclein. Alpha-synuclein is a very interesting protein. On the left side of the screen here, native alpha-synuclein. This is just a bit of a cartoon about this. This is the shape of a protein can adopt. And this is kind of slightly just representative. It's not the exact shape of it. But this protein, which everybody makes, native as in it's just within us, all our cells in our body, particularly neuron cells, have this, and it's important for release of vesicles, in this case neurotransmitters. And it's got a normal role, production and breakdown or recycling and reproduction of this throughout the life of the neuron. Alpha-synuclein is very important. Now, the first gene identified to cause Parkinson's disease was a mutation in the alpha-synuclein gene. That was identified about 30 years ago, that there are certain mutations in this gene that'll lead you to get Parkinson's disease. So then they started studying this, and they found that this mutation makes it more likely that alpha-synuclein will aggregate. So instead of being just, this protein here just doing its normal purpose, and then when it reaches, it's been gone through a couple of cycles, it gets broken, a couple of uses, gets broken down and recycled and so on. Instead, it starts clumping into what's called oligomers and then fibrils, and they are poorly digested. Instead of the native alpha-synuclein, which your nerve cells can digest within them with their enzymes inside them, instead it becomes indigestible, and then it accumulates. And that's what this cartoon shows here. In this green neuron, the red stuff is accumulating now to make these accumulations. These are called Lewy bodies. So the Lewy bodies, which are the main thing we see under a microscope in Parkinson's nerve cells, are mostly composed of alpha-synuclein that's aggregated. And then the kind of alarming thing, or the really fascinating thing actually, is that when the accumulation of this alpha-synuclein, it can spill over from one neuron to the adjacent neuron. And that causes a gradual spread of Parkinson and worsening of Parkinson, that it starts going wrong in a small one, maybe even one neuron. And then it spreads to another 2 or 3, and then from there to 10 or 12, and there from to hundreds. And this is part of the process of progression of Parkinson's. So this insight of the past decade or so about this process seemed to be central in the process of Parkinson going wrong, whether it's genetic, which is one way this can go wrong, but there are other toxins that might influence the folding in a way that makes it aggregate like this and spread neuron to neuron. And we think this may, at least I think, this may be a very important key central step for what's going wrong in Parkinson's disease. So an attempt to stop the transmission or stop the accumulation, or to help the cells break down so it doesn't have as much Lewy body or as much accumulation, is a very important part of Parkinson neuroprotective research in this past few years and going forward. So one of the ways you could try to remove alpha-synuclein is to use your immune system, because it's a protein, and your immune system is very good at recognizing certain shapes of proteins. You can actively immunize. That's to vaccinate somebody to get alpha-synuclein, inject it into their body, and they develop resistance, and then it starts attacking. But that can get out of control real fast. And active immunization of these sort of things has not gone well in research so far. So instead, what the main focus of research now in this line is to do what we call passive immunization. And that's to develop monoclonal antibodies. Those are very purified antibodies that are specific to go after alpha-synuclein when it's misfolded, and to infuse that into the bloodstream, for example, once a week or once a month. And then some of that draws down your alpha-synuclein. Now, as you may or may not know, antibodies don't usually cross into the brain. But if they draw it down from your bloodstream, it kind of resets a balance or equilibration between the brain and the bloodstream, and reduces and draws it down from the brain into the bloodstream, and the bloodstream draws it away in the monoclonal antibodies. That's the theory. And this is a research trial that's going on now. And on the right side, we see a bit, from a phase 2 study that had a few hundred patients. And they followed them for a number of years, and they looked at how they progressed over time. And in the gray, the top line, from your 0 to 0, 1 to 2 to 3, 4, it got worse. They lost more and more points on this scale. Whereas the people who got this immune therapy with the alpha-synuclein drawing it down, they seemed to get worse the first year, but then they stabilized. And they only gradually got a bit worse years later. So there's some hint of a benefit in this. And so this research is ongoing, and we're studying it here as well as other places to see if this is an effective way to slow or stop Parkinson progression. Now I want to switch gears to disease-modifying gene therapies. So there are a couple of different strategies to use a gene therapy to modify Parkinson's progress. You can use growth factors, nerve growth factors, like glial-derived neurotrophic factor, GDNF, is one of the ones that's been most interest in this, or Neuroturin, which went by brand name SIR 120. And the GDNF has shown some signals of benefit in small trials, and it's still being tested some more. It can be infused directly into the brain, or it can be introduced into spinal fluid, or other ways like that. And Neuroturin went through the best and biggest trial so far 3 or 4 years ago, and unfortunately that crashed and burned as well. Or you can use a gene to counteract a defective gene. So there are certain types of Parkinson's that are due to mutation in this LRRK2 gene. And people who have a abnormal LRRK2 can be injected with this therapy that'll counteract that defective gene in a way that's potentially promising. That's for a subset of people with Parkinson's disease. And then you can also replace a defective gene. So there are certain people who have Parkinson's. The underlying cause is mutation in the GBA1 gene, which makes for a certain enzyme called glucose ribosylase. And if, let's say, I think it's some small few percent of people with Parkinson's disease, this is the underlying cause, or they have this mutation. Well, you can give a fresh gene that is functioning more normally and inject that and see if it does a good job. And that's undergoing testing right now. You may have heard of CRISPR-Cas. That's a way to edit a defective gene in a very specific for individual person. So these other things I've talked about so far, that's really giving the same treatment to everybody who fits the criteria. This is very much personalized, selective editing of your particular mutation, your particular synuclein gene, to try to make that right. The problem is you can do that in a certain number of cells, but replacing all the cells and all the genes in the active brain area is an immense challenge. Another strategy is stem cell replacement. So essentially, you're missing millions, actually, hundreds of millions of neurons that are involved in Parkinson's in the substantia nigra area of the brain, for example, and related parts of the brain. They are aging prematurely and they're lost. The number of cells is less. Well, you can inject new cells in that area and you can try and program them or have them replace the function that's missing. Initially, this was tried 40 years ago with adrenal cells, because there are some similarities between adrenal cells and dopamine producing cells. And there were also trials on, so that was about 40 years ago, adrenal cells transplant. And then about 25, 30 years ago, there are fetal midbrain cells. That was early aborted fetuses taking the midbrain and getting those area cells and putting them into adults with Parkinson's disease. And they didn't work out. They actually kind of got out of control, those cells. They were hard to regulate and they just didn't stay well. They aged prematurely. They made bad connections, caused sort of dysfunction. Now, their more modern science is for engineering embryonic stem cells and inducing pluripotent stem cells. That's where you can take even your own stem cells and generate them from your own tissues to turn into neural cells that will produce dopamine and so on. And you can do that and introduce and inject surgically these neurons back into the brain in an area where cells are missing. And you can try and encourage them to make connections and train them to rewire and to restore functions. It's a gradual process, and early research on 10, 12, 16 patients, you see some signs of benefit in the first year or two. And on the imaging scan, we can see survival of these neurons and uptake. So there's some signal of benefit, but what we're really looking for is longer, larger term study to see if these benefits are real, if they're holding up. So in summary, on this brief overview of neuroprotective research in Parkinson's disease, I could say there's some partial victories in neuroprotective research in Parkinson's disease. We talked about recital and aerobic exercise being things already on the books that we recommend. And then, unfortunately, a number of failed trials of things that look good on paper and test tubes. And then the strategies that are currently ongoing and showing some promise. We talked about manipulating the cell environments, antioxidants, for example, drawing down synuclein protein with monoclonal antibodies. We talked about gene therapies to counteract or replace the defective gene. We talked about replacing the dying neurons with stem cells. And those are all ongoing research efforts. And I did show you how to find those sort of trials if you were interested in participating in one of those sort of research. So that's 36 minutes. I didn't want to stop, give at least 20 minutes for question and answers. So do I turn it back to you, Anuba?
Yes, thank you, Dr. O. We have some questions that people posted online when they registered, and then I'm hoping some people will raise their hands. We have a question on here as well. So I'm going to dive into it. The first set of questions had to do with lithium and gut health, and what's the research on that?
Oh, yeah, two interesting questions. So first is about lithium. We'll talk about that, and the second one is about gut. And there's lots of things we could talk about that. But let's start with lithium first. Lithium is an ion. It's like calcium or sodium or something like that, and which is important for at one level for neural excitability, and by using lithium, you can alter your excitability of neurons, for example, in a way that so that's one level. Now, when you do that, it was discovered many decades ago that lithium is quite an effective treatment for bipolar disorder by adjusting the nerve excitability, for example, in that condition. In a similar time frame, a lot of research on animals and models has suggested lithium might be neuroprotective. And it's been tested in a number of diseases. ALS, there's a big research study on ALS, has been looked at in Alzheimer's as well, and in Parkinson's disease. And unfortunately, has crashed and burned in the human trials of all those neuroprotective diseases. So while in test tubes and in theory, lithium is an interesting molecule, in practice, it doesn't seem to have a disease protective effect in intact humans. Now, the gut health, let's talk about that. So as you know, people with Parkinson's disease the neurons in their gut are affected. And this leads to constipation is a very common thing, but other motility difficulties and so on. And if we look at the gut under a microscope, people with Parkinson's disease, we can see loss of neurons there. Now, that's one fact. Another fact, the GI microflora, as you know, you've got billions of bacteria in your gut, particularly your colon, and they have an important role. I don't think it's possible to survive. It wouldn't be possible to be without these things because it's an important part of digestion. They're important. It's just microbacteria, microflora in your gut is just part of how it is. It's just part of nature. Now, there's healthy and less healthy bacteria. For example, there are Vibrio species that reduce sulfate, and we think that's not healthy to have more hydrogen sulfide in your bowels. And it appears that there's more of those bacteria in people with Parkinson's disease. Fact. Now, chicken and egg thing is the problem that your motility is reduced because your neural cells are reduced, and therefore your turnover, and therefore your bacterial environment is different, so therefore there's going to be more of those particular bacteria? Or did you, some bad luck at some stage, get more of these bacteria and they start affecting your bowel in a way that caused the damage and ultimately the Parkinson's disease? Now, that's a bit speculative. I think it's a bit easier for me to believe the first theory rather than the second, that the altered bacteria are the cause of Parkinson's disease. Now, there is some research. Can you change the bacteria flora? And you can. Probiotics, there's ways of introducing bacteria, by mouth, pellets, capsules, things that'll populate your GI system with more normal kind of bacteria population, we would think. And there's some indication that, some people feel better. They have less constipation, for example, or they feel better, feel like they walk better, and so on. The research is kind of ongoing, and it's certainly hard to prove a neuroprotective. I don't think it's a theory or a roadmap for to get from a disease modification by changing your microflora that's going to help your Parkinson long term. It may help you feel better in some ways. So I'm not dismissing the research, but it's and a number of scientists are looking at this, and great, happy and delighted they are. But it's in process. So some patients ask me, well, am I going to recommend it? And so it goes like this. It might help, but the evidence is just not good enough that I usually don't recommend stuff that might help. If I was to recommend everything that might help, I'd be giving you, 20, 30 things, including a few years ago, I would have given you a shark fin, or various other things, or blue algae, because there was some evidence that it might help. But as we definitely found out later, no, it doesn't help. So I don't usually recommend stuff on the basis that it might help. I wait for better evidence, and that may be missing the boat. Some of those 20 or 30 things actually will help when we know the truth. But anyway, we can talk about that's philosophic more than anything else. But go ahead and ask more questions.
Okay, yeah. So there was a question on probiotics. So thank you for answering that. Somebody's asking about benefits or risks to using B1.
That's a vitamin B1? Yeah. I mean, you need vitamins. That's why they're called vitamins. And there are a number of different ones. There's, obviously A and B and C and D and K and blah, blah, blah. And among the Bs, there are, multiple different ones. And they each have an important role. Every vitamin has an important role in the body, and you don't make them. You make many of the important chemicals in your body, but you don't make vitamins. So you have to get them usually from your diet. Now, if you have a good, healthy diet with a good variety of foods, and you absorb healthily, then you should be getting all your vitamins. We're evolved that way, that's what should happen. Of course, some people don't have particularly healthy diets, or there may be some other reason, including the GI, that they don't absorb things quite as well. And so you could make a case for taking supplements. There's not an across-the-board deficit of B1 or any other vitamin in people with Parkinson's disease. And there's not an across-the-board recommendation to take B1 supplementation. To me, there's some evidence that taking B6 and folate and B12 is not a bad idea if you're taking a lot of levodopa. And I'll tell you why. Because levodopa chews up the metabolism or the use of levodopa uses up certain numbers of those vitamins, B6, folate, B12, and if you're taking a lot of levodopa, you may be using up a lot of your vitamins. You may need to take a supplement to stay ahead of the game. But B1 specifically, I don't think there's particularly good evidence for that. Again, everybody should get all their vitamins. It should be in their good, healthy diet. If there's any much doubt about it, take a multivitamin. That's my usual recommendation if you're not sure about your diet or your absorption.
Okay. What about the utilization of lixisenatide to slow progression? And are there another GLP-1 similar that could be used?
Yeah, no, that's a great question. So GLP-1 drugs, everybody's familiar with, for the weight loss, initially for diabetes control, semaglutide, those sort of drugs. And they are fascinating drugs about the biochemical effects of those and the health benefits of those. And so there has been interest in research on semaglutide and GLP-1 agonists for multiple neurologic conditions, including Parkinson's disease. And at least two of them have, I think maybe three different versions of the GLP-1s have been put to a pretty rigorous testing. And one of them, I'm sorry, I had a cold a couple of weeks ago and still a little bit of cough going on. Sorry. So exenatide was the first one that was put to rigorous testing, and it didn't show any benefit. Now, that was only 6 months out. But the people got the real deal, and the people got the other had no difference. Randomized control, good quality trial. So it certainly doesn't look promising for exenatide. Then lixisenatide was put through an even better study. There's 150 patients or so in France, and it followed for, I think it was 6 months, could be a year or something like that. And there was a little book, shown on the screen, a little bit of benefit, and it did reach statistical significance. And that's a hint, and that's somewhat optimistic and promising enough that we should look into it further. But it was only one study, and it kind of counteracts the other study, which was negative, and now you got one positive, one negative. Is it real or not? It'll have to be followed up before. We'd certainly hope for bigger benefit if it was real. We certainly want to see longer term benefit. So we're kind of in a position we would have been with rasagiline, that there's some signal of benefit, but the signal of benefit, frankly, was larger and more compelling, and with multiple studies in the case of rasagiline. So exenatide is not there yet, or any other GLP-1 yet. So we're not recommending it. And I wouldn't take it, because there's so many ramifications of taking a medication like that. There may be some of them good, some of them bad, but it's just not, clear enough that, it's one of those 20 or 30 things that might help. But you mean, which of them? There's no rationale for picking one more than another. All right, I think I'll stop there and take the next question.
Mm-hmm. Well, this question about high dose thiamine for Parkinson's disease and also nicotine having a neuroprotective effect or not.
So thiamine is B1, so that a high dose of this in Parkinson's, no, I don't believe, I'll have to look at it again, but I don't believe I've seen any compelling evidence in humans that randomize to high dose vitamin or thiamine versus not. If anybody has, again, you're going to see a lot of stuff, individual anecdotal experience or test tube work or other things like that that might suggest one thing or another. But when I'm talking about research clinical trials, randomized trials with people with Parkinson's disease, half of them getting high dose thiamine, half of them getting dummy drug, and watch them for a minimum 6 months, ideally 12 months, preferably 18, 24 months, showing a benefit of thiamine. I don't believe anything like that has been done or shown compelling, so we're not recommending that. Nicotine, fascinating. So people who smoke are less, well, fewer people with Parkinson's disease have. There's been a suggestion that smoking is neuroprotective for Parkinson's disease. That's probably not true. But the epidemiologic studies, if you look at the incidence of smoking in people with Parkinson's disease, people who don't have Parkinson's disease, you'll see that people who are smoking are less likely, there's association between smoking and less future Parkinson's disease. If you look at the smoking rate of people who have Parkinson's disease, people who are not, you see a clear-cut difference in a way that might hint that smoking is protective. Now, to do what we call a prospective study, you'd have to get, hundreds of people who are age, I don't know, 40, let's say, who had never smoked. Half of them would start smoking, half of them not smoking, and see, the 1 or 2% would get Parkinson's in just normal course events. If the smokers randomized that got less, and the ones who were randomized, then we'd say, wow, looks like smoking is neuroprotective. Now, of course, smoking is toxic in many other ways, so we never recommend smoking anyway. But there are other reasons. So anyway, based on that research, and the people said, smoking, and damaging your lung, how do we get to nicotine and take that in some way, subcutaneous or pills, something like that, and see if nicotine can be neuroprotective. And that research has been done. And it also has been done to search for a symptomatic benefit. And there's nothing there. So nicotine is not a promising thing. If smoking is associated with neuroprotection, there's no reason to think it's just the nicotine, frankly. I mean, people think cigarettes, nicotine, but there's lots of other stuff in cigarettes. Tar, obviously. Nobody's suggesting that's a good thing, but there are many other chemicals. When you burn tobacco, you're inhaling countless neurochemicals. Some one or two of them might even be good. Or some people think there's something about Parkinson's personality or predisposition that also influences one's behaviors, including risk-taking behaviors and smoking. I can't remember what the theory was of the logic, but anyway, it may not be a cause and effect thing in that way.
The current political administration and government, is that affecting funds for Parkinson's research? Someone has a question.
So be kind of careful here. I don't want to misstate, but I'll try to be, informative. Federal funding for research, federal funding has been very generous research. Now, there is a little bit of an argument. You could say, they're really heavy funders of HIV and breast cancer research and, even pretty heavy funders of Alzheimer's research and, some diseases like Parkinson's could feel like, oh, we don't get a fair shake, blah, 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 we never have, blah, blah. Or you could say that. And then in different, time periods, the government is more or less generous with the NIH budget. And let's put it this way, I don't think there's any sense that current federal government or administration wants to, do a lot of, expensive research. And biochemical research is expensive, and clinical trials are expensive. They also have reduced the indirect funding, which is, I can explain, the importance of indirect funding for the infrastructure, the staff, the IT, the security, all the things that have to happen to allow clinical research to happen. And that's more expensive in some places than others. That's just the way it is. And the federal government has cut down a lot on that. So that means a lot of research has to stop. Now, it's not specifically Parkinson's research, unless you want to do Parkinson's research that involves any words about diversity or inclusion or blah, blah, blah. That would attract attention and probably get defunded. But if you're doing standard Parkinson's research, it's not being targeted to be cut. But it's just across the board, they're cutting on indirect funding and they're being more. And there was big, long delays in approval of research. They kind of essentially put a halt on review of research and funding requests. Now they're getting to it, and the process is getting up and running again. So that's a bit of a roundabout answer, but certainly it's not been a freely flowing funds for Parkinson's research, right?
We'll be getting some questions that are not directly relevant to neuroprotection. So, we're going to leave those out for now. And, please speak to your provider about those questions. There's a question about vagus nerve stimulation and if that helps.
No, I don't believe so. It was a different question about neuroprotection for vagotomy, which is a very interesting thing. This was an interesting observation that people who had a surgery that cut their vagus nerve, there are fewer of those got Parkinson many years later. And it raised the question whether some of the GI stuff transmitted along the vagus nerve to the brainstem might have been a part of Parkinson's disease. Vagotomy is rarely done surgery anymore, but it was an interesting observation. Now, the vagus nerve stimulation is helpful for epilepsy. and I think for some mood difficulties and so on. But it doesn't have a role in Parkinson's disease, I believe.
What about DBS, TMS, HIFU? Are those neuroprotective or only symptom relief?
You're really symptomatic. Uh, there's a couple of researchers on the East Coast, Vanderbilt, I think it was somewhere else, who have a theory that DBS might be neuroprotective by altering the nerve activity in a way that doesn't stimulate and therefore less excitotoxicity and therefore less progression of nerve deterioration over time, and you need less medication. And it's not a crazy theory. It's just it's a little bit out there. And we've been doing DBS for 20 plus years, and if it's neuroprotective, it's not dramatically so. And HIFU, I don't think there's any reason to think that could be neuroprotective. No.
Okay. Somebody's asked about memory improving exercises and if those may have neuroprotective effect.
That's also a really good question. And like the physical exercises, it's even more difficult to do a research of the nature you describe. Because if you were to get 100 patients with Parkinson's disease and you're say, to have them do Sudoku, 5 times a day, blah, 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 and the other half, we don't do any, particular mental exercise, just do your normal stuff. Would they do it? Would they engage in an intellectual, stimulating way? And so that sort of research has not been done in that sort of way. Now, you and I think, at least I think most of us think, well, it's good to use your mind, including your cognitive and intellectual function, to maintain those habits of, flexibility and just to maintain those intellectual skills. So we think it makes sense. We're a bit skeptical. I'm a bit skeptical that there's any particular exercise like crossword puzzle, Sudoku, even those sort of things that are usually mentioned. When we've discussed this at research meetings and so on, I think most of us think it makes most sense to be involved with social conversation, and that's what we're evolved for. And an analysis in the course of, discussing with each other about what's going on in our families, what's going on in the community, what's going on nationally, internationally. It's a good, flexible, relating to people in that way is a good mental engagement that I think is good. So that's why I think it's very important to be socially active, despite Parkinson's disease. Conversations, I think they're probably the best mental exercise. That's not research proven. It's just kind of what I think common sense, and many of my peers think the same thing.
Mm-hmm. That's great because, the Total Care program offers a community and lots of ways to connect. So that's good.
That might be a good way to finish up then. I've got a patient at 3 o'clock. I may have to jump.
Yes, we are right here. Thank you everyone for your participation. Please do fill out the feedback form so we know to do more of these later. And Dr. O, any last minute thoughts that you may have before we end?
Well, thanks for the invitation and thanks for your interest in being part of it. And thanks for, continuing to push us forward, both by encouraging us, questioning us, participating potentially in research studies. This can't happen without people with Parkinson's disease wanting it to happen. So we appreciate your ongoing interest in that.
Thank you very much, everyone.
All right,
Goodbye.
take care. Thank you.